Our first session will be an overview of the basics of adaptive platform trials and suitable questions for an APT, presented by Dr. John Marshall. Dr. Marshall is a professor of surgery and critical care medicine at the University of Toronto and a senior scientist in the Shing Knowledge Institute of Unity Health Toronto. He is the rounding and current chair of the International Forum for Acute Care Trialists. He is the Canadian principal investigator for the REMAP-CAP trial and co-chairs the R&D Roadmap Committee of the Clinical Characterization and Management of COVID-19 of the World Health Organization. He is an associate editor of Critical Care and a senior editor of Critical Care Medicine. Please help me welcome Dr. John Marshall to the podium. Sarah, help me with your slides.
Well, thanks very much, Sabrina, and thank you, Andrew and Benita, for the invitation to be part of this meeting again this year. I'm really looking forward to the day. So I was tasked with addressing the question of the basics of adaptive platform trials. And as I thought of it, it struck me that there are a number of concepts, overlapping concepts that are kind of included in the phrase an adaptive platform trial. And to try to clarify them best, I think it's worth having a look at how each of these has evolved over time. First of all, let me provide my disclosures. A major disclosure is an academic one. My major conflict is an academic one. I'm really kind of passionate about the concept of integrating research into clinical practice, and perhaps sometimes blinded by my enthusiasm for this. But I want to go back to the history of clinical trials and some of the key elements that are incorporated into this most recent iteration of an adaptive platform trial. Trials go back probably thousands of years, but the popular view is that the first clinical trial was this one. It was conducted by James Lind, who was a ship surgeon in the United Kingdom. During the 18th century, there was a real problem when sailors were on ships for a prolonged period of time. They would develop scurvy, and nobody knew what caused scurvy or how best to treat it. And so Lind came up with the suggestion of taking 12 sailors who were on this ship and randomly assigning them to a number of different potentially therapeutic interventions. Some of these are pretty awful. 2 people got sulfuric acid, 2 people got vinegar, 2 people got seawater, 2 people got barley water, 2 people got cider, and 2 people got oranges and lemons. And as it turned out, over the next few days, the 2 sailors that got oranges and lemons started to improve to the point that they began looking after the other sailors who were on board the ship. And without knowing what the cause of scurvy was, this trial had provided evidence of an effective therapy for it. And this is the key element in a clinical trial, is the random assignment of participants in that trial to an intervention or one or a choice of 2 or more interventions. So moving forward to the 20th century, randomization serves to recognizing that in any population there's a tremendous amount of variability, and that variability may impact how a patient responds to a treatment. What happens with randomization is it increases the probability that that variability will be equally distributed between the 2 groups that are being studied, and the only systematic difference is the intervention they're receiving. And because of that, we're able to make conclusions of causation and establish that relationship between the intervention and the outcome that we're measuring. So this was introduced by Lind. Obviously, another threat to the conclusions one draws is the potential for bias in a trial. You may decide to select patients who are more likely to benefit from one therapy than another, and you may adjudicate outcomes differently. So Bradford Hill really introduced the concepts of strategies to minimize bias in a clinical trial by introducing blinding of the intervention and allocation concealment, which means that an investigator could not predict which arm a patient was being randomized to. And that's usually done by just having variable block size for the interventions. But with this is the template for a contemporary clinical trial, and there have been tens of thousands of such trials done. And it really is the basis for our making decisions about what works and what doesn't work in clinical medicine. So this is the situation schematically and slightly out of focus. You take a group of patients with a disease, you randomly assign them to one of two different interventions. It might be a treatment, a control, might be two different treatments. You administer the intervention and then you observe the participants. And at the end of that period of time, the outcome that you're looking at, if it's differentially expressed between those two groups, you can confidently say it was likely the intervention that was responsible for that. So a conventional study puts its focus on the intervention. So in order to do that, I'm going to use an example of a study that we recently did through the Canadian Critical Care Trials Group, where we asked the question, can probiotics reduce rates of ventilator-associated pneumonia in mechanically ventilated, intubated, critically ill patients? So in order to do that as a conventional trial, we had to figure out the event rate. So what was the probable rate of developing ventilator-associated pneumonia in that population of patients? We had to anticipate what would be a treatment effect, what was the size of treatment effect that we would want to see. And having done that, we could calculate a sample size for the trial. But there's a lot of other things that go into developing a clinical trial. As anybody who's ever done one knows, you have to develop the protocol, you have to develop CRFs, you have to develop a database, you have to establish contracts with your sites, you have to recruit the sites. There's a ton of paperwork and that sort of thing, long before you actually start the clinical trial. So this was a CIHR funded clinical trial. It was led by Jenny Johnston and Deborah Cook for the Canadian Critical Care Trials Group. So here's the assumptions that were made at the baseline is that we would have about a 15% rate of ventilator-associated pneumonia. We wanted a 25% relative risk reduction, and we want 80% power and an alpha of 0.05. Now, I'll come back a little bit later to that alpha of 0.05. It's a number pulled out of a hat, and the person who popularized it, Ronald Fisher, said it's the biggest mistake he ever made in his life as a statistician. But it is the touchstone that we all cling to in making decisions about causality. So that's 2,650 patients had to be recruited to this particular clinical trial. It ran for a number of years, and when we concluded the clinical trial, sadly, there was not significant evidence that probiotics made a difference. Now, this is not, we tend to talk about trials as being negative. I would not interpret this as negative. To me, this says that that particular probiotic is unlikely to benefit patients. And that's useful information to know because it's something that we used intermittently. And now we know that we probably weren't helping patients, probably weren't harming them. But it was an awful lot of work, very expensive. And once the trial's complete, it just evaporates. There's no legacy. You've got a whole network of people recruiting to the trial. You've got all of the infrastructure, and it's just shut down and rebuilt the next time you conduct a trial. So that's where a platform trial comes in, because a platform trial shifts its focus. It doesn't focus on the intervention, i.e. probiotics, it focuses on the disease. And any disease may have multiple different interventions that can be effective. So in a platform trial, we're studying the disease. And so that disease might be ventilator-associated pneumonia in a population of patients who's at risk of that. And there's many, many different ways we might be able to reduce the risk of developing pneumonia. We could do it with probiotics, as we did in the Prospect study. We could do it with perhaps prophylactic antibiotics, perhaps topical non-absorbed antibiotics, perhaps positioning, elevating the head of the bed, putting the patient on their side, perhaps physiotherapy, an aggressive physiotherapy course. Perhaps the problem is buildup of biofilm on the endotracheal tube and some model of either a tube that was resistant to that or change in the tube could make a difference. So there are many, many different things we can study. And a platform trial, by studying the disease, allows us to study those things either simultaneously or sequentially. And what's particularly attractive from an investigator point of view, suppose you say, well, I think the idea that changing the endotracheal tube, I think that's a really, really dumb idea. We're not going to do that. Well, you don't have to do it. You can study probiotics, you can study prophylactic antibiotics. As an investigator or investigative site, you take those aspects of the question that you think are most relevant to your particular concerns and study them. And you could bring in new ideas over time. If another potential prophylactic measure comes up, you can add that to the platform as you go forward. I want to just introduce a couple of terms here because this is really a platform upon which multiple trials are being run. And we use the term domain to describe an area of therapeutic intervention that has multiple different options. So antibiotics might be a domain. We could use antibiotic X, we could use antibiotic Y, we could use no antibiotics. So that is a domain. The interventions are which specific thing. So perhaps the antibiotic is a cephalosporin, first generation cephalosporin. That's one of the interventions in the domain. And then we have a regimen, which is the combination for an individual patient of the domains and interventions they were randomized to. So perhaps a little bit complicated, but domain in essence is really the trial on the platform. And the interventions are the things that are being compared in that domain. You can pool all the data across domains, and so you can see interactions, and that's where it becomes useful to think of the specific regimen that a patient received. So here's the way a platform trial is structured. It runs on the basis of a master protocol. That master protocol kind of defines the broad outlines of the population that you're going to be studying, the endpoints you're going to use, basically the PICO, and it sort of creates the structure for the trial itself. For any domain, you have a domain-specific appendix, which may have additional entry or exclusion criteria. It may involve additional data elements that are going to be collected during the clinical trial, but it guides the conduct of that particular domain under the master protocol. And for each domain, you have a statistical analysis plan. So what you see there really are 4 separate clinical trials, all running off a single master protocol, single case report form, single database, and potentially a perpetual structure, so that we could be adding Appendix E, Appendix F, Appendix G in studying this particular broad area. So you can see how that becomes a much more efficient way of conducting clinical trials over time. This is a great review of platform trials written by Scott Barry, Roger Lewis. Barry consultants in the United States have been in many ways the champions of this model. They initially introduced it actually to study early phase interventions for patients with breast cancer. So they used the platform trial to evaluate different therapies and set criteria for graduating those therapies so that they would be studied in a phase 3 clinical trial. But this outlines some of the differences. A traditional trial, single agent, platform trial, multiple agents in a heterogeneous population. Duration of a simple trial till the sample size is recruited. In a platform trial, it's potentially perpetual. It can go on forever. And this is one of the huge challenges, is how do you fund something like this? It doesn't fit with the classical funding models we have. Multiple treatment groups in a platform trial. Stopping rules are actually specified for the individual domains. You can use additional elements, and I'm going to get on to the concept of adaptive designs in platform trials, because a platform trial doesn't need to be adaptive. The adaptive is a separate concept, but platform trials are ideally situated for adaptive designs. Perhaps one of the most important things about platform trials is there's often multiple sponsors for them, multiple funders, multiple sponsors over time. Okay, so I'm going to use as an example the REMAP-CAP clinical trial. REMAP-CAP stands for Randomized Embedded Multifactorial Adaptive Platform Trial in Community-Acquired Pneumonia. This was a trial that was imagined after the H1N1 pandemic in 2009, because we realized that you couldn't launch a trial during a pandemic and expect to recruit an adequate number of patients. You really had to have the trial ready hopefully recruiting in advance of the pandemic need, and then simply adapt it to meet the needs of the pandemic. So we set up REMAP-CAP to be ready for the next pandemic need, and as everyone is fully aware, unfortunately that need arose in 2020. It actually arose before we were quite ready to respond to it, but that's what life's like, and we had to deal with that. So REMAP-CAP, as it was adapted to study patients with COVID-19, is a modular trial. We had this trial studying patients with community-acquired pneumonia. We had maybe 120 patients at the end of 2019, and we very quickly switched it over. We had a pandemic protocol that defined how the trial was going to change. Our endpoint changed with the pandemic. And then it was a question of figuring out what we were going to study. Well, platform trials were beautiful for that because obviously, questions of antibiotics, corticosteroids, antiviral agents, those were sort of the first tier of questions that people began to think of with COVID-19. But very quickly, people said, well, maybe immunomodulation may be beneficial here. So we added a domain that looked at a number of different immunomodulatory agents that were available. So interferon beta, anakinra, which is IL-1 receptor antagonist, IL-6 receptor antagonist. Vitamin C, there was reason to think that that might be beneficial in ARDS, and there was a trial going on studying vitamin C, so we added it to the platform and federated with that particular trial. Very early became obvious that patients with COVID-19 developed coagulopathy and clotted readily. So antiplatelet agents and anticoagulations were added to it. Statins seemed to make sense both based on previous work and on the potential anti-inflammatory activities. We realized as we were going that we didn't really have a domain studying mechanical ventilation, and that was pretty important, so we added that. The SARS-CoV-2 binds to the ACE receptor on epithelial cells, so we thought, well, maybe if you block that binding, we can prevent that. So we brought in the ACE2-RAS domain. So you can see the trial is adapting and is modular. It's adapting as we learn about the disease. We can bring on new interventions that speak to that evolving body of knowledge. So this is where REMAP-CAP currently stands. We're close to 15,000 patients worldwide. We've had about 10,500 randomized with severe COVID-19. And it has enabled us in a period of really just a matter of a few years to draw some important conclusions. First, that hydroxychloroquine is harmful. Lopinavir, ritonavir is not beneficial. It's probably harmful. Corticosteroids, on the other hand, were beneficial. Anti-inflammatory agents, IL-6 receptor antagonists, and it didn't matter which one, were beneficial for patients. On the other hand, IL-1 receptor antagonist in this population wasn't beneficial. We saw a dichotomy in the response to heparin, depending on how severely ill a patient was. Saw no benefit for convalescent plasma, no benefit for P2Y12 inhibitors, no benefit for vitamin C, no benefit for ACE2-RAS inhibitors, but a very strong suggestion of benefit for patients who receive treatment with simvastatin. And so these have all been reported. We have 14 publications so far and are continuing to put out data on some of the other interventions, which include, for example, ivermectin is one. We've studied corticosteroids in community-acquired pneumonia, et cetera. Okay, so this is, the acronym highlights some of the features or the aspirations of the trial. The E is that ultimately this will be embedded in the electronic health record so that data collection can happen almost automatically once a patient has been identified as being eligible for the trial. I want to just focus a little bit more on some of the adaptive aspects of it. I've showed you one, that we can change the interventions as the trial goes forward. But an adaptive trial is a trial that has built into it an a priori plan to change as the data begin to accrue or as things are learned about what's being studied. So it's a design that allows modifications for the trial or the statistical procedures without undermining the integrity. As you know, if we were just to say, let's have a look at the data and oh my gosh, we see this particular thing, so let's change the trial this way, you're introducing a huge amount of bias. If you specify these things in advance and you say, if this, then we'll do that, if this, we'll do that, and you develop a whole algorithm about how the trial will respond to the data it's accruing, you don't run the same risk. And you can really adapt many different things. I mean, adaptation is not unique to a platform trial. Frequentist trials often adapt based on sample size. If the rates of disease are less than we thought, we're going to increase the sample size, for example, if the event rate is lower. But in an adaptive trial, you specify these things in advance, and that's what gives it the benefit. So here's how it works in the trial. You begin data collection with the initial allocation and sampling rules. You analyze the available data. You determine, has the stopping rule been met? And that may be stopping the domain and reporting it. If it is, then you stop. If it isn't, you revise the allocation based on that a priori algorithm, and the trial keeps recruiting, and potentially that can go on forever. So one of the really neat features of adaptive platform trials is the potential to change the randomization proportions during the trial based on the accruing data. And it's called response adaptive randomization. And the idea here is that for a patient in the trial, if the trial is seeing that one arm seems to be doing better, ethically, it makes sense to randomize more patients to that arm, and statistically it is possible to do that through a pre-specified change in the randomization proportions. So this is how that would work in a hypothetical trial where you look at 3 different drugs compared to usual care. You randomize in a 1-to-1-to-1-to-1 ratio at baseline. You conduct these analyses, and as you go forward, you see that usual care and drug B seem to be doing somewhat better. So more patients receive those, and by 700 patients, it's a trial of usual care versus a single drug, but the single drug seems to be doing better, and you've reached a conclusion. And at that point, all patients receive that trial as part of standard care. So that's a very, very attractive concept because it really marries clinical research and quality improvement, and integrated into the healthcare system, you could be constantly improving and updating the care you provide patients based on the information that's coming through the clinical trial. Okay, so I've gone through a few things in clinical trials and how adaptive trials deal with them. Randomization, adaptive trials are randomized. Blinding, we don't in REMAP-CAP use blinding because of the number of interventions, but we use hard outcomes, mortality and requirement for organ support. There are adaptive elements to it, but the final thing in REMAP-CAP is the statistical framework we use to analyze the data. And there are 2 different approaches. The frequentist approach, which is the most commonly used one, where you're evaluating the probability of the data given the hypothesis, and the Bayesian approach, where you're evaluating the probability of the hypothesis given the data that are occurring. They're kind of complementary approaches, but they involve different assumptions, and they, to my mind, they reflect a different perspective, and I've become increasingly enthused by Bayesian thinking because it is what I do as a clinician. I think in a Bayesian framework. So frequentist trial estimates the baseline rate of the primary outcome, the change resulting from the intervention, and the confidence in the resulting estimate. And that's how you calculate a sample size for a clinical trial. In our trial of probiotics, you can see the difference here, and I showed you that it was going to be 2,650 patients. We recruited that. What about the outcome from that? So we saw, using the frequentist model, no significant difference between the 2 groups in the output, which was mortality. And in a frequentist model, what you're estimating is the probability that the result you saw in that trial is as extreme or more extreme than you would see if you did the same trial over and over and over and over and over again. And as I said, arbitrarily, the value selected to determine significance is a p-value of less than 0.05, but that just means there's less than 1 chance in 20. It's kind of hard to get your mind around this because the assumption here is that there's not an effect. That's the basic statistical assumption. And the analysis is trying to reject the possibility that there is no effect, but that it might actually be worse. So the other model is the Bayesian model, and this was Thomas Bayes, who's a clergyman who came up with these ideas, and they really only emerged after his death. But the basic concept here is that in trying to interpret any differences we see in life, we don't start as a blank slate. A frequentist approach assumes we know nothing. We're just going to assume there's no difference, and we're going to test the hypothesis that this difference we see was likely to have been a result of the intervention. Bayes says, well, that's not how life really works. I mean, we start with prior assumptions about things, and you can incorporate that into the analytic framework that you use. When you run a clinical trial, you can look at, as the trial goes forward, the posterior probability that there's a difference between the two different things that you're seeing. And so in a Bayesian analytic model, you are looking at the distribution of probabilities for the two different groups that are, or two or more different groups that are there, and looking at the probability that that distribution is occurring randomly. I won't go into too much more, but in REMAP-CAP, what we are looking at is the odds ratios for the outcome of, in the COVID-19 iteration, it was organ support-free days over 21. In non-COVID, it's survival at 90 days. And we're looking at the odds ratio of a difference. We conclude superiority once the posterior probability in a Bayesian framework is 99% that the odds ratio is greater than 1.2. Equivalence, when there's a 0.9 probability, the odds ratio is between 0.8 and 1.2. And inferiority, if there's a posterior probability of 99% that the odds ratio is less than 1. So this isn't specifying a sample size. We're simply running the trial until we cross one of those stopping boundaries, and at that point, we conclude that domain. So, and what this actually is saying in a Bayesian framework is we're looking at the probability after the trial has been done that the therapy is effective, which to me is a much more intuitively attractive approach to thinking about outcomes. So as I mentioned, in a platform trial, you have multiple domains. They're analyzed independently. You stop when you've met an a priori boundary for benefit, harm, or equivalence, and then you simply report those as you would for any randomized controlled trial. And potentially you can use those results in treating subsequent patients who are recruited to the trial. You can stop for other reasons. We stopped several of our domains for operational futility, for example. The statins one, rates of COVID-19 were dropping, and we still hadn't crossed a boundary, so we stopped for operational futility. And one can say I mean, it's fair to say we did not support a conclusion of superiority. We also didn't really meet our criteria for equivalence there. So this is how our endpoint was shown in publications. We used an ordinal scale, which was just the days over 21 that a patient was free of organ support. If they were on organ support, it was red. If they died, they got a -1. And so we're just comparing visually here, comparing the two different groups. And you can see these are the differential results with heparin in critically ill versus non-critically ill patients. It's not a great way of presenting the data, and I think it's somewhat hard to look at those and say, oh yeah, I see the difference there. Certainly a Kaplan-Meier curve is easier to interpret. So I wanted to say, finally, a few things about the operations of a platform trial. REMAP-CAP at its peak was operating in 364 sites around the world. We have sites on every continent, but very heavily weighted towards the UK, where clinical research is much more integrated into care than it is in other countries. We have multiple different partners and sponsors that are involved in the clinical trial, and they come from around the world. None of them include trials groups. We've got multiple different funders for the trial. Canada, the CIHR and the SPORE program have been instrumental in funding this, but we have private funders as well involved. Its governance is a little bit complex. It's run by an international trial steering committee, which consists of about 50 or 60 people. We have a kind of methods component where there's a design team that develops the domain-specific protocols, statistical analysis committee that does the analyses, a data coordinating center that brings data from multiple different data providers. I think we had 7 at one time, based in multiple different regions, and they all went to a central data coordinating center and a data safety monitoring board. National regional centers and national regional appendixes. So Canada is one particular center, multiple data providers. We have domain-specific working groups. Each of the domains has a working group that designs the protocol and the statistical analysis plan. And then we have a series of operational committees. So things like a prioritization, patient and family engagement committee, and that sort of thing. And they all have representation on this International Trial Steering Committee. It's a collegial thing. It's fun. We have Zoom calls at 3 o'clock in the morning, depending on which area is kind of coordinating it, and that gives us has given us enough coordination that it's worked. Okay, so I was asked to address the question of when is a platform trial design appropriate. I think a couple of issues here. If the focus is a disease, thank you, with multiple treatment options, it's good. I think there has to be a long-term commitment to collaboration. It's particularly useful for research networks because you can bring together multiple different questions on a single platform, and it's a potentially more efficient way. It's also really good for international collaboration because it gives both mentorship and education, but also autonomy, and as I said, ultimately in embedding research within care. Just very quickly, I mean, we're running it in Southeast Asia through a registry. We've had over 1,000 patients recruited from Nepal, Pakistan. They lead the ivermectin domain of the trial. We've been holding, over the last couple of years, international meetings on trying to build better collaboration and understand how trials work together, including this one just a couple of weeks ago in Dublin, where we're trying to bring together platform trials from around the world to figure out how we best create an ecosystem based on the platform trial model. And we're working with the World Health Organization to try to build a globally federated trial of optimizing oxygen in hypoxemic patients, particularly in low and middle income countries. So I think the important thing here is that this is an opportunity to embed research in clinical care, and that 10 years from now, we can imagine a world where we have multiple platforms running in a particular care area, and patients, rather than dealing with uncertainty, we simply randomize patients and we continuously learn and improve that care as we go forward. So I'll stop there and be happy to take any questions.
Okay. Thank you, Dr. Marshall. Hi, everyone. My name is Kevin. I'm a research coordinator here at the Upstream Lab, and I'll be facilitating the Q&A for all these sessions. So if you have a question in this room, please raise your hand, and Antonio or Sabrina will be giving you a mic to answer so that you can ask your question. And if you are asking questions online, please enter through the Zoom chat, and one of our staff members will read it out loud for Dr. Marshall.
And if you want to challenge anything I said, please do. This is an evolving field, and I don't want to pretend that this is, gospel. This is just my perspective on it.
Okay, so does anyone have any questions?
I'll start. Thank you so much, John. Amazing overview. I had a question about the decision-making about response adaptive randomization. This, in our experience, this is one of the areas where there's a lot of different viewpoints. And I'm just wondering, with an international group of people, how do you come to a decision about that, the pros and cons of it? One of the issues has been just the technical challenges of implementing response adaptive randomization. That's sometimes been part of it.
Yeah, it's a great question. It's the one big bugbear, and I think it's something, frankly, we have not done very well in REMAP-CAP. Our it seemed like a really good idea, and our initial response adaptive randomization allowed a randomization up to a 90-to-10 ratio, so it's a 9-to-1 ratio. Big problem with that because you don't have as many people to compare once you do it. If anything goes wrong, you run into problems, and it unblinds it. People say, look, you know, every patient's getting heparin, or every patient's getting this something fishy going on here. So we've changed that, but we ran into a few problems, I think, with response adaptive randomization. It was, you know, it was driven by algorithms that said, based on the accruing data, this is what the assignment proportions are going to be. We had some problems with data providers where we were slow getting the data pooled, and it turned out that things had changed and that we ended up randomizing more patients to interventions that weren't doing as well, which was problematic. So we still are using response adaptive randomization in the trial, but we've changed the proportions down to, I think, 0.7 rather than 0.9. And I think it's a big area. One of the things you have to do is you have to have contemporary controls. So we do it in 2-week buckets. The controls for a given patient are patients who were recruited in the same 2-week period. They may not have been actually in the domain. There's some really neat stuff going on in the statistics where you can borrow. It's called dynamic borrowing. You can borrow from other domains. But I think response adaptive randomization is theoretically really attractive. Practically, there's some big pitfalls that we've got a lot to learn about.
Thanks. I'm very new to adaptive platform trials, so this may be a basic question, but I work in a disease where the outcome is something that's achieved at 18 months or 2 years. And you know, REMAP-CAP, the outcomes are very short term. So I wonder your thoughts on using platform trials for something like where I work, tuberculosis, where basically our outcome happens 18 months to 2 years after randomization. Is that a disease that you should consider platform trials, or you shouldn't? Do you need to have a short-term outcome?
So I would think the critical factor in considering whether you do a platform trial is there one thing you want to study, or do you kind of see a spectrum of interventions that might be beneficial? If you think there's a spectrum, I think that that's an ideal situation for a platform trial. You could do it at 18 months. I mean, during the pandemic, we changed our primary endpoint to organ support-free days over 21 because we wanted to speed up the process of response adaptive randomization. Our actual endpoint was, you know, our peacetime endpoint was 90-day mortality. But you could imagine, for example, looking at something in tuberculosis where you took an early marker of, you know, burden of the microorganism or something like that, that predicted that late outcome and used it. But there's no particular reason why you can't have a 10-year outcome for platform trials. It's going to take a lot longer, but it's the same as in any other kind of trial. A 10-year outcome takes 10 years to get. I think there's a question here, James.
Thank you, John. That was really, really great overview. Question also has come up around regulatory approval, and when you do a study like this, if it's not the sort of standard RCT, placebo-controlled, blah, 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 and that you're using sort of Bayesian analyses, what has been your experience with, you know, getting no objection letters and clinical trials approval, and then subsequently, once you demonstrate a benefit, how do regulatory agencies look upon the results? Like how do they consider things like an OR of 1.2 to be a sufficiently solid benefit to give regulatory approval, as this may have relevance to funding treatments, et cetera, long term, and getting approval that could be covered by private insurance, et cetera?
Yeah, no, it's a great question. I think it's important to recognize that this model has been around for a while. It's just kind of become more popular as a result of the pandemic. The FDA has guidance for platform trials and for adaptive designs and for Bayesian analyses. They're fully accepting of these as models. Our endpoints are probably slightly more rigorous than a typical frequentist trial because of the odds ratio that we're looking at. I'd have to defer to a statistician as to whether that statement is reasonable or not, but there's no particular reason. I mean, regulators basically say that's your risk. You say what the value you want, and you're taking the risk. We're not going to tell you what to do there. But I think the other things, a number of the domains of REMAP-CAP were regulated, Health Canada regulated domains, and it was very, very smooth working with them. They were incredibly responsive during the pandemic. We could talk to them at night, you know, to get approvals and that sort of thing. So. And remember that there are some very active platforms in the pre-registration space in clinical research. I-SPY2, which is breast cancer, GVM-Agile, which is glioblastoma. So regulators are quite familiar with the design.
That's all the time we have for questions for this session. Thank you again, Dr. Marshall.